Now, let's look at the communication between neurons by looking at the different types of neurotransmitters which help propagate information through synapses. Glutamate is the primary exciter in the brain. It plays a key role in the synaptic plasticity, learning, and memory. It binds to inotropic AMPA and NMDA receptors and modulatory metabotropic receptors of the glutamate metabotropic receptor, which increases neuronal excitability and synaptic strength. Clinically, excessive glutamate activity is associated with neurotoxic effects or excitotoxicity and is implicated in degenerative diseases such as Alzheimer's and amyotrophic lateral sclerosis, or ALS. GABA is the primary inhibitory neurotransmitter in the brain. It counteracts excitatory signals and helps regulate neuronal excitability. It has an important role in providing feedback inhibition, which is fundamental in physiology for generating oscillations that encode sensory stimuli into memory and learning. GABA binds to GABA A receptors, which are ionotropic, chloride channels, and GABA B receptors, which are metabotropic, leading to hyperpolarization of action potentials. Clinically, dysregulation of GABA is associated with epilepsy, anxiety disorders, and movement disorders like Parkinson's disease. Dopamine, released by the ventral tegmental area and substantia nigra. Dopamine, secreted by the ventral tegmental area and substantia nigra. Dopamine is involved in the reward circuit. It plays a huge role in motivation and motor control. It modulates several brain functions, including mood regulation and cognitive processes. Binding to D1-like excitatory and D2-like inhibitory receptors, it influences neuronal firing and synaptic transmission. Abnormal dopamine levels are associated with Parkinson's disease of low dopamine and schizophrenia with dysregulated dopamine pathways. Dopaminergic projections to the medial septum or diagonal band of Broca from the ventral tegmental area are involved in regulating hippocampal theta rhythms, which are crucial for memory, learning, and spatial navigation. Serotonin, secreted by the Raphi nuclei, is important in regulation of mood, appetite, sleep, and cognitive functions. It is crucial for emotional balance and circadian rhythms. It binds to various serotonin receptors, influencing neuronal excitability and neurotransmitter release, playing a more modulatory role. Clinically, imbalances in serotonin levels are linked to depression, anxiety disorders, and some forms of migraines. Now, looking at synaptic plasticity, which is the ability of synapses to strengthen or weaken over time in response to increases or decreases in their activity. This is fundamental for learning and memory, and is seen primarily in Hebbian learning mechanisms, where neurons that fire together wire together. Synaptic plasticity is a crucial way for the brain to adapt and networks to remodel for new stimuli and situations. Long-term potentiation is a long-lasting enhancement in synaptic strength following high-frequency stimulation of a synapse. Long-term potentiation is associated with an increase in AMPA receptors on the postsynaptic membrane via increased vesicular fusion from the enhanced excitation and increases neurotransmitter release. NMDA activation is critical for initiating long-term potentiation. Long-term depression often involves reduction in AMPA receptor density or changes in intracellular signaling pathways that lead to decreased synaptic efficacy. It is involved in synaptic pruning and refinement, and alterations in long-term depression processes are associated with developmental and neurodegenerative disorders, including autism spectrum disorders. Another key concept to define within the central nervous system is synaptic connectivity. This can be convergent or divergent, where multiple neurons can converge onto a single neuron, and where one neuron can fire to stimulate many neurons. Now, looking away from the central nervous system, let's take a delve into the peripheral nervous system. The peripheral nervous system connects the central nervous system to the limbs and organs. It is divided into the somatic nervous system, or SNS, under voluntary control, and the autonomic nervous system, or ANS, which is involuntary. The somatic nervous system contains sensory afferent neurons that carry information from sensory receptors to the CNS, and motor efferent neurons that transmit motor signals from the CNS to skeletal muscles or effector glands. It controls voluntary movements and reflex arcs, where sensory neurons convey information such as touch, pain, and temperature to the CNS, while motor neurons stimulate muscle contraction to produce movement. The autonomic nervous system controls involuntary bodily functions and is divided into three divisions: sympathetic, parasympathetic, and enteric. The sympathetic nervous system arises from the thoracolumbar region of the spinal cord and utilizes a two-neuron pathway of preganglionic and postganglionic neurons. It prepares the body for fight or flight mechanisms with responses during stressful. It increases heart rate, dilates pupils, relaxes airways, inhibits digestion, and mobilizes energy stores. Norepinephrine is the primary neurotransmitter of the sympathetic nervous system. It is released from postganglionic sympathetic neurons and acts on adrenergic receptors in various tissues, binding to alpha and beta adrenergic receptors to mediate effects such as increased heart rate, vasoconstriction, and bronchodilation. Reuptake of norepinephrine is taken into the presynaptic neuron by the norepinephrine transporter for recycling or degradation. The parasympathetic nervous system originates from the brainstem and sacral spinal cord, also using a two-neuron pathway, however with longer preganglionic neurons. It promotes rest and digest activities, so it slows the heart rate, constricts pupils, stimulates digestion, and conserves energy. Acetylcholine is a primary neurotransmitter in the parasympathetic nervous system, especially at neuromuscular junctions. Acetylcholine is a primary neurotransmitter in the peripheral nervous system, neuromuscular junctions, and it is also found in autonomic ganglia. It mediates muscle contraction and regulates autonomic functions. In the neuromuscular junction, acetylcholine is released from motor neurons and binds to nicotininergic receptors on muscle fibers, leading to muscle contractions. While in autonomic ganglia, acetylcholine binds to nicotinic receptors on postganglionic neurons. Acetylcholine is rapidly broken down by the enzyme acetylcholinesterase in the synaptic cleft to terminate its action. The enteric nervous system consists of a divergent network of neurons embedded in the gastrointestinal tract, regulating digestive processes independently of the central nervous system, including peristalsis, enzyme secretion, and blood flow to the gut.